So good evening, everyone. Thank you for joining us. Bienvenue à toutes et à tous. I'm Ruth Slack, and I'd like to welcome you all to the third evening of Brain Health Awareness Week, hosted by the University of Ottawa Brain and Mind Research Institute. And again, the objective of Brain Health Awareness Week is to raise awareness about brain health and mental health knowledge that is important to empower us to take care of our own health. So this week's program will focus on keeping tabs on COVID, the latest research advancement, and the impact on mental health. So you may ask why we're looking at COVID-19 in Brain Health Awareness Week. Well, COVID-19 has had devastating effects on the brain, on brain health, on mental health in our community. You can just imagine the isolation throughout the course of the pandemic. The stress and the anxiety has really taken its toll, especially on our most vulnerable, including seniors, and we heard about that on Monday, also with our youth. So it's a topic that really needs attention. So tonight we will discuss how we can keep tabs on COVID, the levels in our community, how we can prevent the spread of COVID in our own homes. So if somebody contracts COVID, how do we prevent the entire family from getting this? And what is the impact of COVID, or what impact does COVID have on mental health, with a focus on our healthcare workers who have worked tirelessly throughout the entire pandemic. So now it's a real pleasure to turn this event over to our moderator, who really does not need an introduction, Dr. Alex McKenzie. Dr. McKenzie is a pediatrician at CHEO. He served as the CEO and director of the CHEO Research Institute for over 10 years. He was the vice president at Genome Canada. His research specializes in molecular genetics related to pediatric diseases. And he really has contributed so much to the academic and medical research community in Canada and internationally. So thank you, Alex, for being with us. And we really look forward to having you moderate this.
Okay. I'm probably one of the most immoderate moderators going, but thank you for those very kind, perhaps overstated words about my contribution. Thanks very much, Ruth. Back at you, by the way. At any rate, okay, so yes, as we're going to hear from Dr. Phillips later on this evening, it's been an incredibly stressful time, exacting a toll from us all, the various levels, but also it's been a remarkable time scientifically. We've made huge advances. Never has a virus been so intensely studied in the history of the world, really, from a genetic epidemiology and an immunological point of view. And we have two snapshots of that this evening with both Tyson Graber and Roger Zemek. And then we're going to have a strong finish with Dr. Phillips talking about the actual mental health toll on some of the medical residents in our fair city. But I just, I don't have much to add to that. It's been a remarkable last 2 years to sit and watch it all develop. Glad it's over, and I think we really are beginning to say we're heading towards endemic versus a pandemic, but huge strides scientifically. When I originally, my son told me that basically I should start looking in wastewater for CoV-2 as it was shed by the intestine into the stool, and this would be a good way of tracking the community numbers. Like any good academic, I immediately took the idea for my own and ran with it and contacted Rob Delatello or vice versa, and we started going. But soon I found that they needed somebody with actual expertise. I did not read the fine print, somebody that knew a bit of science around this, and they were looking at RNA. And so I, in some desperation, reached out to my friend and colleague, Tyson Graber. And I just want to say, Tyson is, by his own admission, description, did a PhD in Ottawa and went to Nahum Sonnenberg and others in Montreal, and was a neuroscientist. back in the day, and actually had a great PNAS paper in 2013, reactivation of stalled polyribosomes in synaptic plasticity. That is really some serious stuff. Was a neuroscientist, but now, as he says on his webpage, he does molecular scatology. He finds out why and how ribosomes do what they do, and dealing with middle age are the three things that he mentioned in his bio. He's a great talker, a great speaker, and we're glad to welcome Tyson to give us an overview of wastewater epidemiology. Make us laugh, make us cry.
Thanks, Alex, for that very kind introduction. As he mentioned, I am not a virologist by trade, so getting into this was a little new for me. However, I did do a postdoctoral study, postdoctoral time with a colleague, Tommy Alain, who is a virologist. So I was working on viruses a little bit, so I know a little bit about them. But this venture into wastewater, like for a great many scientists across the country and indeed the world, it opens up new avenues of discovery. And together with Rob Della Tolla, you're going to see slides coming up here once in a while. And together with Rob Della Tolla, who's actually a wastewater engineer, and Alex McKenzie, who's a pediatrician and scientist, we put together this, or helped put together this great team in Ottawa. And I say a team because it's not just the three of us, it's a very large team actually pictured here at the wastewater treatment plant in Ottawa. And it's very interdisciplinary. So we have epidemiologists on the team, we have medical officers of health, associate medical officers of health, we have basic biologists like myself, cell biologists, we have clinicians, we have wastewater engineers, the whole gamut. So all of these people were required to put together a project like this and to indeed keep it going throughout this pandemic. And so tonight, I'm just going to do a very brief overview of maybe some of the mechanics and the biology behind what we're doing. And this was essentially my knowledge of the wastewater network when we started all this over a year and a half ago. Basically, if I'm here at CHEO, go do your business, and then wastewater, i.e. poop, coming from toilets and other excretions, go through a pipe and they end up in Ottawa at a wastewater treatment plant. And so it's a single wastewater treatment plant in the east of Ottawa. And this services over 90% of our population. So the vast majority of population within the city of Ottawa are sewered, which makes it a very good sampling location. There's just a single plant to sample from. So logistically, it's relatively straightforward. And a technician at the plant on a daily basis now for over a year and a half goes down every 6 hours, takes a sample, puts it in a fridge, and then we merge 4 samples a day to create a 24-hour composite sample. And that composite sample is taken to the lab downtown at the University of Ottawa and Dr. Della Tolla's lab, and it's analyzed for COVID-19. So the virus that causes COVID-19 specifically. And so what you're seeing here in this slide is what we're publishing on a daily basis openly. So this is through participation and collaboration with Dr. Doug Manuel, an epidemiologist, and a big believer in open science. And so the idea that we should be, this data should be public facing and hopefully almost in real time. And you'll see in the orange line is the trend over the last year and a half, the entire study period from the beginning of the pandemic until today. That is a measure of COVID-19 or SARS-CoV-2 viral load in Ottawa through wastewater. And the green kind of teal color is actually the percent positivity. So the case percent positivity. So how many people who go in and get tested for COVID clinically, how many are positive? And so that percent positivity correlates quite well with the wastewater signal. And moreover, we found that in certain circumstances, the wastewater signal actually leaves the clinical data by at least a few days, if not longer. And so this is an early warning signal, if you will, of what's happening to SARS-CoV-2 viral load in the city. And so I'm going to go through 3 questions in this presentation, or hopefully I'll get through these 3 questions. Number one, what do we measure? So I say COVID-19 or SARS-CoV-2 viral load. So what does that mean exactly? What are we measuring? Well, we're measuring the virus that causes COVID-19. So this is SARS-CoV-2. This is a space-filling model done by a quite famous now medical illustrator, Veronica Falconieri, who's done a lot of work for the popular science media. So you'll find her work in Scientific American. Please go and look at her work. It's great. This is SARS-CoV-2, an envelope virus, meaning that there's a lipid envelope surrounding it, protecting it from the environment, but it's actually quite fragile. So it's kind of made from the same lipid material as our cells in our body. As soon as you expose it to the environment, it can break apart. In orange here is what we're actually measuring. So this is the genomic RNA, or the material, the nucleic acid material, genetic material that encodes all the proteins required to make a SARS-CoV-2 virus. And we're actually measuring that. And so it's thought that this virus particle, as we get infected, it doesn't replicate in the gut. However, we tend to shed pieces of it, or indeed actually intact particles in our feces. And that is excreted and it's deposited into the wastewater. From multiple studies around the world, we know that it's not infectious once it gets to the treatment plant. It's somehow degraded to a point which we don't really know how degraded it is, but we can measure the genomic material associated with that virus. So how do we do that? Let's step back a bit and think how amazing this is. So every day at the wastewater treatment plant in Ottawa, that single wastewater treatment plant, the equivalent of 150 Olympic-sized swimming pools flows through, the volume of water flows through that plant every day. That's 350 million liters per day going through that plant, okay? And this is those 150 Olympic-sized swimming pools on your left here. And we sample from that 150 Olympic-sized swimming pool volume, 350 million liters every day, 0.25 milliliters. So what is that? That is essentially a raindrop of material. So a drop in the proverbial bucket. We take this and we sample this. It's not as clean looking as this when we get it. So in the wastewater treatment plant, it's actually a pretty simple process, pretty physical process where the wastewater from from households and industry is pumped up to a higher level, and then it's put through the plant with gravity flow, essentially. And you'll see number 2 on this slide. We're actually taking the primary sludge. So what is primary sludge? Very simply, it's wastewater put into a large vat, and then all of that solid material is settled down through basically time and gravity. And then we collect that. So that's the primary sludge. That is a mother liquor. And we found through a couple of publications that most of the SARS-CoV-2 RNA signal, that signal from the virus, is concentrated in that fraction. And so we sampled that fraction, that primary sludge. This is what primary sludge looks like, probably not too surprising. It's mostly water, 90% water, but largely fecal material and other organic matter coming through the wastewater system. So again, how do we exactly detect that RNA? Okay, so we use a classical methodology in molecular biology, and you're probably familiar with it now with the pandemic. The same technology that is used when you go for a COVID-19 test to assess whether or not you're infected with SARS-CoV-2. This is the polymerase chain reaction. So not going to go into great detail, but essentially this is kind of a molecular bloodhound. It can look in a whole variety of a whole population of RNAs. And so you can imagine in the wastewater matrix, we have a lot of our different RNAs. So it's not just SARS-CoV-2 RNA, it's all sorts of RNA from plant material, from us, from humans, from animals. We all have a lot of RNA in our cells. Different viruses are RNA based. And so all this RNA we extract through physical chemical means, and then we convert it into DNA. And so with this DNA, we can then amplify it with this polymerase chain reaction methodology. And essentially it goes in, we use kind of molecular beacons or molecular bloodhounds that go in and target specific sections of DNA. And so these specific sections target SARS-CoV-2 RNA, specifically the N, the nucleic capsid region of that genomic RNA. And then we copy that over and over and over and over again. So in fact, we copy it 10 billion times before we actually get a signal that we can attribute to the viral load, SARS-CoV-2 viral load in the wastewater at any given time. And so when we started this, my thinking of the wastewater system was as just poop flowing through pipes, wastewater flowing through pipes. Now my thinking as a biologist is, well, there's RNA flowing through these pipes. And what does RNA and DNA mean to a biologist? It means information. There is a whole crap load of information running through our pipes on a daily basis that we can extract and understand. And so this gets us to a point, but where, how much time do I have? This gets us to a point where we're actually measuring that signal. And it's not just whether or not SARS-CoV-2 is there or not, so a binary yes or no answer. We're actually measuring the magnitude of that signal, so the viral load. And so you can imagine that things like dilution effects come into play. So if there's a lot of rain happening in Ottawa at any given time, that's going to dilute out. That's going to increase the flow going through the plant. And it's going to dilute out or dilute down the SARS-CoV-2 signal. And so we have to correct for that. How do we do that? We look for something that is present in all of our feces. This is worldwide. It's pretty ubiquitous in urban areas, especially. We look at a specific virus, a plant virus. It doesn't affect us, but it affects pretty much everything we eat. So processed foods, hot wings on a Friday night, you've got a lot of this pepper mild model virus flowing through you, and you're depositing it all the time in wastewater. And so we actually normalize, use this as a fecal content marker and normalize our SARS-CoV-2 signal to this pepper virus. And so this is how we get a consistent, robust signal that we can measure as a magnitude and follow over time. And so we can compare signal right now in November with the signal that was that we got out of the system in last July, and it's comparable because we're normalizing to this pepper virus. Okay, so just as an example of how this works, on the top is a raw RNA signal, and then on the bottom is a pepper virus normalized signal. So you can see slight differences here. And so we think that this is better or more accurate, a more accurate representation of SARS-CoV-2 viral load in the city at any given time. So how is this information, how is this data, how is this technology being used to improve public health? Well, scientists, and in particular epidemiologists, so that speciality of science where these epidemiologists are looking at how disease propagates through populations. This is a valuable tool for them. So clinical testing is great. We absolutely need it, but there's inherent biases in clinical testing. You might be biased towards one particular cohort. You might be biased towards one sociodemographic cohort or population. And this is kind of a little bit of a great equalizer, this wastewater-based epidemiology. And so it actually is passive surveillance. So we're collecting this information from everybody, whether they like it or not. And so we can get a great deal of information out of this and correlate that with the existing clinical data. And this is a valuable tool for epidemiologists. Moreover, this methodology complements existing clinical surveillance. Why? Because it provides an independent measurement from clinical data. So this is a concept in epidemiology called triangulation. So you see this triangle right here. Each vertex or corner of this triangle represents a different method, so an independent method. So you can imagine that wastewater information can be one vertex, clinical case data, so your nasal pharyngeal tests can be one corner, and the third corner could be hospitalizations. When you have all these independent measures together, you get a better picture of the reality that's happening in your city, in your neighborhood, of what the disease is doing, what the prevalence of the disease is in the population, and how it's moving through the population. Okay, and maybe running a little over time, so I apologize here, but just one kind of use case here, again, for the city of Ottawa. Back last fall, when cases were kind of low, we kind of opened up a little bit again. And the province said, oh no, let's tap the brakes on that and let's put in a stay-at-home order. Part of the reason why we did that was that, or why they did that rather, was that wastewater in Ottawa at least was starting to go up. Clinical data was not. So clinical cases were still holding steady, but wastewater was spiking. And our mayor suggested that lockdown wasn't necessary. Our premier disagreed, and in that case, at least, the premier ended up being right. A few weeks later, our chief medical officer of health, Dr. Vera Etches, who's an ally and really enjoys the wastewater-based epidemiology project and the data that it's generating, she tweets out that watch out, Ottawa, wastewater is going up. And then at this point, a few weeks later, towards Christmas, clinical cases were also going up. And so this is an example of kind of forewarning that can, I'd like to think, it empowers a community. It brings data into the hands of the population. Again, independent measurement from the clinical measures. And so this, I think, is extremely important and kind of a new avenue of public health that hasn't really been explored up to this point. I'm going to skip this. I'm going to mention the Ontario Wastewater Surveillance Initiative because this entire talk was about Ottawa, but this methodology has exploded, not only in the province, but across Canada and across the world. So over 60 countries now, 3,000 sites sampling across the world, looking at SARS-CoV-2 and wastewater. And here at home in Ontario, what most of you are probably not aware of now is that more than 70% of the Ontario population is actually being monitored in this way now, using methodology that started here in Ottawa. And so over 133 sample applications across the province. This includes over 82 central wastewater treatment plants that encompasses all the major urban areas in Ontario, together with smaller upstream facilities, so long-term care homes, neighborhoods, correctional facilities. 1/4 of the First Nations in Ontario are now being monitored using wastewater surveillance. And the vast majority of public health units are now using this data on at least a weekly basis to inform them as to what's happening with SARS-CoV-2 in their communities. So altogether, it's really been a great project to be part of, together with Alex and Rob, and it's really, I think, a poster child of what basic science can do in these situations. And I think that it's also an example of how well teams can work together when they introduce kind of these interdisciplinary notions into their research. So I thank you for your time.
What a great job, Tyson. They originally asked me to do it, and I said, no, get Tyson. And you did so much better job than I would have done, Tyson. Amazing. Plus the Prezi video, which is like, I've never seen it. It's very cool. There may be questions about how you access it.
Yeah, well, it's free as long as you don't mind having this little watermark.
Okay, it's very cool.
Yeah, free advertising for Prezi.
Oh, okay, but great. Diana Wilkinson has a question about RNA stability, but Diana, if you can just hold on to the end, we'll just go to Roger now, and then Jennifer, and then we'll have a panel discussion and air all the questions. And so Roger is an old. Thanks again, Tyson. Awesome job. Roger's an old dear friend. He's got a great CV. He did applied mathematics at Yale University, medical school and residency at Columbia before doing Peds, Emerge, and McGill, and becoming an immigrant from the South. And he's been a practicing ER physician at CHEO, I guess 15 years now. Roger, I lose track. You can correct me. Senior scientist at the CHEO-RI, clinical research chair in pediatric concussion at the U of O Brain and Mind Institute, director of our Clinical Research Institute at CHEO, scientific director for the Evidence to Practice program, vice chair of the Pediatric Emergency Research Canada PERC network, led the largest prospective concussion study in the world to examine predictors of concussion syndrome after concussion. Has some very interesting work coming out of that in the near term about exercise post-concussion, has started a sort of, do I dare say American style, but sort of a concussion clinic outside the walls of CHIO to look after this huge unmet need called 360 concussion care, along with the previous Governor General's extended family, if I could say that, at any rate. And so that's a great model of sort of exporting research in healthcare from the walls of CHIO. And now he's masquerading as an immunologist, if that's not enough. Roger, welcome, and we look forward to hearing your talk.
Well, thank you, Alex. It's going to be hard to follow Tyson and all those amazing double entendres that he threw in about a poop. I was just thinking this would be so entertaining for a bunch of middle school boys to hear. They would be learning and laughing all along.
The jokes never stop.
Exactly.
In that area.
Well, I will do my best to do that. One may ask me what experience I have studying viruses. I would say, as someone who's worked for 20 years, primarily as a pediatrician and primarily in the front lines of pediatric emergency, I think I've caught every seasonal virus there is. So that in that sense, it makes me an expert. So I am pretending to be a virology expert with my the true experts on the call today, but I am a scientist, as Alex says, and I love to ask questions that hopefully have some relevance. And my team and I, when SARS-CoV-2, which has caused the COVID pandemic, started, we were wondering, how does this really affect kids? Because a lot of the studies that were done were really focused on children, excuse me, not on children, but on adults, and in or particular demographics such as healthcare workers or the families of healthcare workers, and we weren't really understanding how this was going to affect the normal population. So we designed a study to look at who gets immunity, how long does that immunity last, how can it be spread within households. So I'm the one member of the team presenting you today, but it's a true team effort. My co-PI and I've known for over 20 years. We actually did our fellowship together back at Montreal, is Mala Bat, and she's an associate professor of pediatrics and works with me in the emergency department at CHIO. And we brought on the real expert, Marc-André Langlois. He's a professor and a Canada Research Chair in molecular biology, and he is the true brains behind the science. So we assembled a truly great team who I'll introduce at the end as well. And when we started this, as I briefly introduced, we knew that likely the number of infections in kids was really underestimated. Who could be tested was very strict in the beginning because they really wanted to save testing supplies. We also were only testing people with symptomatic, and we knew that asymptomatic spread was important. I can anecdotally say in the emergency department, I've seen dozens of cases of COVID that I've treated, and at least half of them had nothing to do with the classic signs or symptoms. It could have been the child who broke their arm, and I fixed their arm and put it back straight and put on a cast. And then a day or two later, I got a call from public health saying, were you hopefully wearing your mask and your gown and your gloves because your patient that you treated for their broken arm was positive as well. So we knew that the understudied groups included children. It included the people that were non-hospitalized and walking around in the community. And it was also knowing that a lot of the tests were done in the traditional nose test, and there may have been other ways, especially looking backwards, are there better ways to know if someone had been infected? And while Tyson and Alex and his team have talked about the looking in the forward, what we're doing is the converse of that. We're looking backwards. Was there something that may have given us a clue that you had been infected with the virus that causes COVID? And so the questions that we wanted to answer was, if someone in the household gets it, you're living under the same roof, are there other people? How likely are you to spread it? Is it spread different if it's kids or adults? There was a lot of budding evidence in the beginning saying kids don't even get it. There may have been thoughts that kids, if they get it, don't spread it. That was based on conjecture rather than data, and we wanted to confirm with what was the truth or a best estimate of the truth. And then the question was, How do kids develop their immunity through antibodies, and does it differ? Are some people more or less likely to develop antibodies, or does it change based on how old you are or how sick you were in the beginning, or whether you're a boy or girl, a man or woman? And then lastly, how long do the antibodies confer protection for? I won't talk about that today because we're still in the process of analyzing that data, but I'll talk about some of the first critical questions that I can share with you, some of that preliminary findings. So this was not, although we have CHIO scientists as a member of the team, we also had members of the Ottawa Hospital, and this was truly a citywide, similar to Tyson's surveillance of the city. This really was a citywide surveillance. So we partnered with all the testing centers. This is a picture of the Brewer COVID assessment center, and I'm sure some of you on the call are familiar with those tents and that lineup. And we also partnered with all the major emergency departments through the city. So pretty much any test that could have been done in the city, you were eligible to be enrolled in our study. And we even reached out, and this is actually one of the things that was really interesting. This is the first time I've ever used Twitter. I'm not a tweeter myself, but we did partner with our CHIO Research Institute Communications Department, and we were able to develop this infographic and sent it out through CHIO's and the University of Ottawa's and the CHIO Hospital's tweets, and just kind of reached out to the community and said, hey, if anyone in your family has tested positive for COVID, come find out, because a lot of families may have had just one person in their family tested. So it could be a multi-generational household and one of the parents was positive, but they therefore were all under quarantine. They didn't want to put their child through the painful procedure of being tested. So they decided, oh, we'll all just quarantine and we won't necessarily all undergo the somewhat uncomfortable nose testing. So the way to enter the study was you had to live in a household which had at least one person under 18, and one person in that household had to have COVID positive by that PCR test that Tyson just explained to us about. And so this was not based on, oh, I had systems consistent with it, or I had what's called an antigen test, which is maybe not be as accurate as the PCR test, but this was an official through the city testing system at one of the testing centers or emergency departments positive for COVID-19. And then the second thing was, if you were in that household, since we wanted to look at how things spread, you had to have not just the one person enrolled in the study, you had to have at least one other person in that study enroll. And overall, you had to have at least one kid in the study and at least one adult. What was interesting and what was amazing, people were so interested and willing in participating. And we had over 650 total participants, almost 200 households, and for almost 80% of the entire for 80% of the households, the entire family signed up. So there were households with, multiple kids, 5 or 6 kids, all signed up, both parents, and even other generations who are still living under that roof. So it was tremendous buy-in. And I said, I'm going to thank the rest of the study. This really shows about the power of building a prospective clinical research team, because it was our nurses and our coordinators who really did the hard, heavy lifting of encouraging and getting parents and kids to come in to CHIO to get their blood drawn and to be tested. This is a test that we only can do by drawing their blood. And what we would do is we would draw their blood. We would take it to the lab of Dr. Langlois, and then he would test for the presence of antibodies. And there are several types of antibodies. There's IgG, which lasts, what we say is a longer lasting one. But we also measure 2 other types of antibodies, IgM and IgA. And then we look to see how long those antibodies lasted. So we did it at the time of enrollment, and then at 2 months, at 6 months, and then we're going to look again at the 1 year. So let me just jump to what you've all been waiting for. We wanted to answer the question of, can kids spread? So if a child had it, we wanted to know how many of the kids spread to someone else in the household. and what percentage of household contacts did they spread to who were previously not known to be positive. They were either not tested or they were negative for their testing. And what we found was it was almost 40% of the household contacts. When a child had COVID, it was spread to 37% of that household. So again, almost more than a third. Then we looked also, what if the index case was an adult? It could be the mother, it could be the father, it could be extended family member, such as a grandparent that's living under that multi-generational household. How many other people in the household get it? And what was interesting, it was even more. It was 57%. When you look at both together, it was about half of household members did infect each other. When we looked at what were the factors that could determine what was more likely or less likely to spread, households that had higher density. So it was the number of people living in a household as compared to the number of rooms in a household. So a higher density was more likely to spread. What was the relationship, as we talked about, someone who's the primary caregiver, such as a mother, if they are the one responsible for doing or one primarily doing things that have to be done, like feeding or changing diapers, more likely to transmission. And again, there was also probably a dose response. And what I mean by that is the more number of people in a household who were positive at the beginning, the more number of cases were likely to become positive in the household outside of that. The other question I want to talk about, this is like behind the scenes super secret stuff. So it's not peer reviewed yet. It's not published yet, but I think it's very interesting. And I wanted to share it because I do think it is controversial and I do think it is important. What we also looked at was we looked at those people who we knew were tested positive for COVID. So they had the PCR test confirmed, they had gone to Brewer, we knew they're positive. There were about 200 participants in our study who we knew had that. We wanted to know, are there any people that just didn't develop antibodies? And what we found was that about 10% of adults and 17% of kids never developed antibodies. This is really interesting and important for a couple ways. What does that mean? It means that people who recovered from known COVID are not theoretically or may not be immune. There are different types of immunity. There are things such as we talked about, the humoral response, which is the antibodies you develop. There's another type of immunity, which has to do with the things called T cells, and also known as memory cells, which also tie into our immunity. And we have partnered and gotten an additional grant to study the T cell function of those people, particularly who never developed antibodies, but we knew did have COVID, to see did they at least have some other longer term protection. But what's really interesting is we don't know if these people are immune, and that may explain why we're seeing people getting COVID more than once. This also may explain why we're seeing continued spread even in people who had a natural disease as well. The other thing that was really important for this study was we proved that kids can actually transmit COVID. We show that they can spread it to about, again, 37% as household contacts. What's interesting about kids is they also go to school. So it may be really interesting to think about that fine balance of where on that Goldilocks balance of not too hot, not too cold, where should we be lying with regard to children? As a pediatrician, and we're going to hear from Jennifer shortly about the mental health impacts of COVID on our community. I think it's so important that kids get back to school, but what we should really do is continue keeping those face masks, even for the youngest children, because we know they can spread, and continue to have, encourage as much vaccination policies as possible to better protect these kids from when they want to do recreational activities in schools for things that don't require or can't allow masks, such as eating in the cafeteria or doing their sports, which are so important. So again, we have a great team of physicians all across different disciplines, from emergency medicine to infectious diseases to statistics, amazing research coordinators and nurses. And we even had a patient representative who gave us feedback on how do we best approach families and communicate with families and design our study based on that. And again, we need to thank our funders. We worked with multiple funders for this, and we also work closely with the public health agency. So it was a true pleasure to work on this study. I think it's still, we're learning what's at the tip of the iceberg, but it's been exciting to study and be a part of coming up with some solutions to this thing that affected all of us for the last 2 years.
Brilliant, Roger. I personally have a ton of questions about particular strain, whether socioeconomic status is involved, whether how you control directionality of infection. But why don't I show uncharacteristic restraint, park those, and then I'll pepper you with them at the other side. But very exciting, great stuff. Not bad for emergency room doctor who's sort of wandered into immunology. Muzzle tough. Great job. And so now I'm delighted to introduce Jennifer, and who we've not met Jennifer, although I do study ketamine and autism to some extent, so we need to talk at some point. But at any rate, Jennifer is a scientist at the Mood Disorders Research Unit at the Royal Institute of Mental Health Research. She has a PhD in neuroscience from the University of Ottawa, and is the director of research for the Department of Psychiatry. It says interim, but I imagine you'll get the gig, Jennifer, whether you like it or not. She's an adjunct professor at Carleton, and she studies the biological underpinnings and consequences of depression using MRI and peripheral biomarkers. And she's testing the efficiency of low-dose ketamine to treat depression in patients resistant to other forms. She has waded into the mental health consequences of this, and it must some days been the days must have been long sometimes, Jennifer, because it's not necessarily a happy or uplifting tale as you delineate the impact on the residents, on the medical residents in Ottawa. So we look forward to hearing you tell your tale. Thanks very much for joining us.
Yeah, thank you so much for having me. I'll just get my slides. Okay. Yeah, so I'm really happy to be here to talk about the mental health impact of COVID-19, particularly on medical residents. But I think what I'm going to say largely, in general, largely applies to most healthcare workers. And we'll talk a little bit about that as well. Again, this is a big collaborative project as well. I'm going to introduce some of the people as we go, but mainly on the last slide to thank and acknowledge collaborators. So we know that the global spread of COVID-19 has led to profound economic and societal disruption with the potential for widespread mental health consequences. What everybody else has mentioned, there's been enormous sort of scientific mobilization to characterize the virus, to develop and test vaccines, but considerably less focus to date has been paid to the mental health impacts, to understanding them, to mitigating against them, especially for certain groups, including healthcare workers. And due to the unpredictable and ongoing nature of the pandemic, medical and nursing staff have really essentially faced sort of chronic stress conditions for months after months after months. And this can have a risk of accompanying mental and physical health consequences. As I mentioned, I'm going to focus on medical residents. Some of you may not know what medical residents are, essentially. This is a part of one of the many steps towards becoming a specialized, independently working physician. So individuals attend medical school for 4 years, and then they engage in residency. And residency is a period of supervised training as a physician. So they're physicians working, but they're still technically in training. And they're specializing in a particular field of medicine. So you may have individuals in a surgical residency or a psychiatric residency or learning to become a family doctor. So the training is very specific to a particular branch of medicine. Medical residents have been working on the front lines in our hospitals throughout this pandemic. It's an interesting time, I would say, for them to be completing this most important part of their training. And as a result of being sort of frontline medical workers, they're at increased risk for negative mental health outcomes. Within Canada, under normal circumstances, rates of depression and suicidal thoughts are actually higher among physicians than the general population. In a pre-pandemic study, so done before the pandemic in 2018, there was a national survey done of physicians by the Canadian Medical Association. And they actually found that burnout, depression, and suicidal ideation was higher among medical residents than among practicing physicians. So there are many factors inherent to medical training, like excessive workloads, long hours, up to 80 hours a week, and lack of autonomy. These things contribute to unintended consequences on medical resident mental health. And we think that the stresses of working during the pandemic may be exacerbating sort of existing challenges. So in order to study the impact of COVID-19 on medical resident mental health, we administered a bilingual cross-sectional survey online. And this was a big collaboration with lots of partner hospitals in Ontario and Quebec, and a social media sort of campaign to have people fill out the survey. Some of you may have done a survey that's related to this, because this is part of a larger or broader survey effort that was led by Dr. Rebecca Robillard from the University of Ottawa School of Psychology, which actually looked at the mental health impacts of COVID-19 on various populations in youth, in essential workers, in people at higher risk of negative outcomes from COVID-19, as well as in healthcare workers. And this work was funded by the Canadian Institutes of Health Research in a grant awarded to myself and my co-PI, Dr. Katarina Nikolic from the Department of Psychiatry. So in all, we had 111 participants who responded to the survey. This included 80% of them were women, so highly biased towards women. And most of the participants you'll see were from Ontario, with a smaller amount from Quebec and then other provinces. In all, we had representation from all 6 years of medical residency and from 29 different medical specialties, with the most respondents from family medicine, psychiatry, internal medicine, and pediatrics. Overall, at the time of survey completion, 57% of respondents reported that in their current work, they had in-person contact with patients who tested positive for COVID-19. So these are individuals who are working directly with people with COVID-19. And we also wanted to know at the start how many of our respondents had a pre-existing mental health disorder. And we found that 36% reported a current mental health diagnosis, and about a quarter were taking psychiatric medication at the time of the survey. In terms of mental health outcomes, 91% of residents reported increase in their stress levels since the onset of the pandemic. In order to assess depression and anxiety, we used validated scales. For depression, we found that 47% of residents screened positive for clinically significant depressive symptoms. This actually means that they may meet criteria for depression. Further assessment would be necessary to make a diagnosis, but that's essentially what that is, screening positive. That's what that means. For anxiety, 57% of medical residents had moderate or severe anxiety, and 16% of residents endorsed suicidal ideations, suicidal thoughts, in the 7 days preceding survey completion. So these numbers may seem really high, but within this sample, actually the rates of depression and suicidal ideation were actually almost identical to those that I just showed you in that pre-pandemic national survey. What may be significant here is that almost all the residents self-reported increased stress since the pandemic onset, and more than half had moderate or severe anxiety symptoms, something that wasn't measured in the earlier national survey. So a lot of us had increased stress during the pandemic. Many of us may have experienced some depressive or anxiety symptoms. What we think might be going on that's unique with medical community here is something called moral distress. So imagine facing a situation at work. You know what needs to be done, but you feel prevented from doing what you think is morally right because of healthcare limitations like understaffing or time constraints or lack of resources. Part of our study, in addition to the online survey, we're also conducting interviews with individuals from the Faculty of Medicine at the University of Ottawa. These include medical students, medical residents, and medical educators, so faculty members that are teaching students and residents. And I have a quote here from a third-year University of Ottawa medical student that provides a good example of a morally distressing situation. So this medical students that were in their last 2 years of medical school, normally they do rotations and they go into the hospitals and they learn there. They were faced in the COVID-19 pandemic with cancellation of in-person clinical work, and a lot of them volunteered their times elsewhere. And this individual volunteered as a COVID screener in one of our Ottawa hospitals. Here's how they described the difficulty of that role. I think it was really hard because you would have pregnant moms who couldn't be in active labor with their partners. And then you would have patients who were palliative and none of the family members could be let in simultaneously. It was really difficult to have to turn patients away because of that. So this is an example of moral distress, a concept that's been borrowed from the military. Feeling unable to do what you believe to be ethically or morally appropriate because of institutionalized obstacles. In this case, COVID restrictions. I think we've all heard stories or seen in the news of individuals who are unable to go in and say goodbye to their loved ones in person, and we're having to do it over FaceTime. So this is a morally distressing situation for the people who have to stop the loved ones from coming and saying goodbye. So to assess moral distress within our medical residents, in the survey, we used the measure of moral distress for healthcare professionals. Overall, the moral distress scores within our survey were actually relatively low. And we saw no differences among residents who had that in-person contact with patients positive for COVID-19 and those who were working elsewhere where they were not in contact with COVID positive patients. But what we did find was that higher degrees of moral distress, when it was present, were associated with higher depression or anxiety symptoms. In the larger survey I mentioned, from which this resident data derives, we actually had a sample of almost 1,000 healthcare workers. So in this sample, we have physicians, residents, nurses, psychologists, social workers, and hospital administrators. They were all administered a large survey, and we were able to statistically look in more detail at the relationship between moral injury, moral distress, and poor mental health. And what we found was that the more exposure to these morally injurious events, the higher the likelihood of somebody developing moral distress. And the more moral distress somebody experienced, the higher they had poor mental health outcomes, elevated stress, depression, or anxiety. So an ongoing area of research now is to understand the factors that may protect against the development of moral distress in the first place and against the development from moral distress turning into some sort of mental health disorder. So what could a healthcare professional do who is experiencing moral distress? There's a lot of, as I mentioned, a lot of ongoing work in this area, but so far the evidence supports self-care, peer support, so debriefing and sharing experiences with your coworkers, and professional mental health support. Last bit of data I'm going to present here, we actually did survey a subgroup of our residents about their mental health service use during the pandemic. This sample includes 51 participants and medical residents. Overall, half of them actually considered seeking professional mental health services during the pandemic. We asked them what would make you hesitate to seek help. And their answers were things like concerns about confidentiality, the costs, worry about stigma, and time constraints. Remember that 80-hour work week, it's very difficult to find time to go have appointments. Among those who did seek out care, 31% of those who considered seeking care actually sought out care. And among those who did, there was significantly more women who sought care than men. The good news is that of those who sought care, 94% found the care that they received helpful. And the type of services accessed were primary care physicians, psychotherapy, physician wellness, or crisis or emergency services. So I think it's pretty evident that there may be an increase in mental health symptoms among healthcare workers. A lot of this work is still ongoing and needs longitudinal follow-up so that we can see where we end up with this. But it does appear that we are coming towards the end of the serious part of the pandemic, at least here in Canada. But history has also shown us that the effects of the pandemic, especially the mental health impact, may be long-lasting. So for example, the SARS epidemic or outbreak in Toronto in 2003, even several years later, 3 years later, you could still see a mental health impact among medical and nursing staff. So a new study that we're going to launch in the coming months is to look at the physical toll of the pandemic to see whether the chronic stress conditions under which healthcare workers have been working actually elicits any physical attributes in the body. So as mentioned in the intro, my normal area of research is using magnetic resonance imaging to scan the brain, as well as taking blood and saliva samples to look at stress hormones and inflammation in the periphery. And we're going to apply these kind of techniques in a study where we're recruiting frontline healthcare workers to try to understand if we can identify any biological vulnerability or resilience factors that help predict or prevent the development of mental health symptoms in the face of this chronic stress or moral distress. And that project's funded by an anonymous donor with funding facilitated through the Ottawa Community Foundation. So something to stay tuned for. Lots of people work on this project, as you see here. Most of us derive from the Faculty of Medicine at the University of Ottawa. We have medical residents who informed a lot on the questions that we asked in this project. We have medical students, educators. We also have some partners at the Canadian Medical Association, and that's about it. So thank you.
Jennifer, a great job. Can I just take moderator's privilege and just, did I catch that if you look at the delta as far as mental health issues pre-pandemic and post, and I guess during pandemic, there at some levels there may not have been as great a shift as one might have anticipated.
Yeah.
Okay.
Yep. A lot of research is showing that. I think there's a lot of conflicting research. I don't have a good solid conclusion for you at this point. Sure. But it's possible that the stress, the extreme stress of medical training, yes, and that some doctors are used to working under may actually be protective, yep, and we may not be seeing the kind of outcomes that you might, yes, anticipate. We might be seeing something different in nurses. Yep. We might be seeing something different in different populations within the medical community, or in different areas, which maybe were harder hit by COVID.
Right. Okay. Brilliant. Okay. All right. Wonderful. And so that is it for three great talks. And just looping back to some of the questions that have been posed, Tyson, I just might, and thank you very much, Jennifer. That was more illuminating and less down and less depressing than I had anticipated. A great talk. Tyson, if we could loop to you and ask, Diana Wilkinson asked a full hour ago almost now, at what rate does the RNA degrade within wastewater?
Very good question. So it could be answered in a few different ways, but I'll give you an easy answer. It does degrade, we know that. If we look in the lab, so we try to recapitulate what might be happening in the wastewater system in the lab, so kind of in vitro. If you take wastewater and then spike it with poop that is from an individual who has had COVID-19, this is being done by a PhD student, so Liz Mercier, and she's finding that the RNA signal degrades quite rapidly over the first few days, so by roughly half. But over time, over 60 days in the beaker at various different temperatures, even at almost room temperature, there's not a whole lot of additional degradation. So question is why? Why is it so stable? Because if you do have some background in biological training, you're always taught that RNA is very fleeting, and that in the environment, and indeed in cells, once they break open, there's enzymes that actually are there to degrade RNA, because RNA actually produces stress responses in cells. And once that RNA degrades, there's no signal left. And so understanding why that RNA might be still hanging around in these environmental samples, they must be in some sort of protected molecular species, and we're trying to understand what that might be. Another part of the question might be how does it degrade in the body? Or what form that RNA takes in the body, and that's another area that is active research. We really don't know how this virus or fragments thereof are excreted in the body through waste. So the answer is yes, it does degrade. At what rate? Probably dependent on at least in the body on physiological constraints, but in the wastewater network, probably dependent on a whole bunch of different components, temperature being one, the various proportions of lipids and proteins there. So it might differ between cities and different regions and different sampling locations. One thing I'll just add quickly is that the signal actually degrades quite quickly after freeze thawing of wastewater. So we found that fresh wastewater, and we
Right.
sample that and then detect it. And then we freeze it down, we archive it, and then we come back months later and then isolate that RNA and then look at that signal again. We see a loss of about 50% again in that signal. So we don't quite understand why yet, but yes, the RNA does degrade. And so over time, we're going to see a loss of that signal.
So if you're freezing your wastewater at home, stop. It's
right.
a general public message. Okay, Marguerite Vanderberg has asked about influenza, and I guess both Tyson and Roger can speak to this. I guess, Marguerite, I think you're asking, are we going to use this wastewater hammer for the influenza nail, Tyson? I guess you mentioned something at the end there.
Yes, so it's anticipated that we can also use this methodology for looking at influenza virus.
Mm-hmm.
The question is whether we can get adequate amount of signal out of looking at RNA-based detection methods. Mm-hmm. And again, PhD student Liz Mercier is looking at this actively. And you know, the problem right now is that influenza is so low prevalence
right.
here in Canada. It's really hard to find a proper wastewater sample with a known amount of influenza in it.
Yeah, but as Roger's about to tell us right now, I think perhaps that won't be a problem for long, Roger?
Yeah, I mean, there's the epidemiological surveillance of an entire city. It's like Tyson and Alex, you are doing through wastewater. There's also the boots on the ground surveillance that we see every day in the emergency department. So we have through our PERC network, the Pediatric Emergency Research Canada, we're doing surveillance. Actually, we're using all 15 sites of Canada funded by Public Health Agency of Canada. And we've even got another grant from Health Canada to look at COVID surveillance across all 15 centers. And as part of that, we are layering on additional respiratory virus screening. So as we all know, there's not just COVID that circulates, there's other seasonal respiratory viruses that can cause fever as well. We call them FRIs sometimes, febrile respiratory illnesses. And in addition to influenza, there's things like RSV. And for those of you unfamiliar with RSV, it's the most common reason for hospitalization in babies. We have had one of the worst RSV seasons in memory. Lots and lots of admitted kids ending up in the intensive care unit, intubated, not with COVID, but with RSV. So we are surveilling not just COVID, but looking at RSV, influenza, parainfluenza. The virus most commonly causes croup. And so those are some of the things that we are looking at through our in-person surveillance as well.
Do we feel the median age of the RSV patients is older than in previous years, therefore it's a more severe illness?
Yeah, so it's still the babies that get the sickest. But what's interesting is normally we say RSV will present like a very bad asthma attack. It causes inflammation of the smallest tubes right before the air sacs, the same
Right.
size as tubes that are involved with asthma. And we basically had a whole cohort of children who didn't really get RSV for that year. So
Yeah.
we're getting almost 2 years worth of RSV. It's still the littlest babies that are the most sick, but we are getting older kids as well, perhaps because they didn't have that natural annual exposure
Yes.
to that
Right.
virus.
Yeah, RSV, a bit like malaria, you need to get it a few times to get a true immunity. So I have a question about ascertainment for Jennifer, Roger, and Tyson. Ascertainment is an issue with any scientific study. Jennifer, I worry, where were the surgeons? Where were the men? How does that sort of constrain your study? And is there something you can do to address that? I mean, it was voluntary. You're just getting people who are coming in. Is there a way of sampling those who are more reluctant?
Yeah, I think there's lots of different approaches that you can take to improve sort of to limit the bias in the sample. Most online surveys actually are predominantly filled out by women. There's a question in the chat as well as to whether or not the physicians and residents who are suffering the most may not have completed the survey. I actually kind of think it might have been the opposite, that people who are experiencing mental health conditions might be drawn to that type of survey and willing to fill it out. So I think it's a great question. These are the caveats we always have to take into consideration when interpreting findings. Our sample is really, really pretty small. I think it's important to look at samples from multiple centers and multiple different populations, and we're still looking at our data. We do have some surgical residents who've done some of the qualitative interviews that's sort of paired to the survey data. So I'm hopeful that we will get a nice representation across specialties. We do have 29 medical specialties in this survey, so there are some surgeons. But yeah, very important questions concerning who's actually doing this.
Right.
They're responding. Yeah.
And Roger's one's anxious that it's the middle-class families in Orleans who are signing in for years, and the harried Syrian immigrants are not being represented in this era of EDI, et cetera. Do you have any sense for that at all in your sample?
It's a great question. We did look at all the different factors. What was great was we did have a nice smattering. To answer your question, though, specifically, have we analyzed to ensure that it was the same proportions as we should have seen normally in the city? I don't want to misspeak, and I don't know that off the top of my head, but I know for a fact it was exactly that situation, Alex. Some of our families would arrive with multi-generational refugees family and 7 kids, and all got tested, and it was really great to see the buy-in of those families. The other thing, as a nice little plug, is the emergency department we just have, and CHIO just got a use of technology. I know unrelated to this, but we have a new iPad with a thing called Voice, which the people actually, it's on a little cart that you wheel around. It's like a little mini robot, and it's kind of that live translator, one language to other language, and it's that great little interaction to really improve communication. And I see that being a great research tool to ensure that we're not limiting people based on a language preference or comfort.
Great. Just to ask the obvious question, Delta, Alpha strain, I mean, one would think that Delta variant would be different within a household than a pre-Delta variant. So what I guess the timeline was all pre-Delta for your study, Roger?
Yeah, it overlapped a bit of both. We did have some Delta. We did measure the variants of concerns. And so we did have some Delta. What was interesting and going forward, which put a bit of a monkey wrench, but again, opens up other opportunities and helps us refocus our priorities, is the vaccination rates. So it's been so tremendous in Ottawa, which is a wonderful thing. As a scientist, it makes it hard because doing the 1-year follow-up, the blood work, looking at their antibodies, it's impossible to know whether the antibody development was then due to vaccine or re-exposure or combination of the two. So we have likely for the 1-year follow-ups will exclude people who've been vaccinated, except for certain groups, which is a nuance, probably too detailed for the purposes of this call, but it is an interesting question on how do you study then the post-vaccine response in people who had natural immunity. Super interesting in the little kids or those who were previously non-responders.
Right. And as we talk about vaccinations, et cetera, just pivoting to you, Tyson, how would the vaccinations mess with a wastewater signal? Do they have, would that be an influence in what you see in there? Is the vaccine mRNA measured by the wastewater assay?
Right. So that's one question we get asked a lot is you know, can you get kind of false positive signal from those who are vaccinated? And the answer, at least the methodology we're using here in Ottawa right now, is no. So we're targeting a completely different part of the genome, so different section of RNA, different code. So we're not, and the vaccines being used now are a completely different area of the genome. And so even if this were to happen, which even if you were to have that RNA present, that vaccine RNA present in wastewater to high enough levels, no, we wouldn't be able to detect it using this. Another part of the question is how do vaccinations affect the signal? You know, presumably with vaccination, you would have less disease prevalence, and therefore wastewater signal would go down. Indeed, if you go onto the public website, you can see that just around the time that vaccinations came in the springtime, when we were seeing surges with the Alpha variant, cases or the wastewater viral load started to go down along with clinical. And indeed, for the past few months now, wastewater signal is extremely low.
I mean,
so that would suggest, you know, correlation, not necessarily causation, but I think we can probably all agree on this, that this is due to vaccination.
And we're seeing just in recent days evidence of standard testing of an upward trend in COVID cases. What does the wastewater tell us in the last?
So the wastewater has been a bit of a roller coaster over the past couple of months. It's been oscillating up and down, not peaking to high signal like we've seen up in the previous waves.
Right.
But there are kind of mini waves.
Yeah.
And you know, trying to correlate that with the case positivity or the number of cases in the city at the moment is difficult.
Right.
I'll have to admit, it's a little bit uncoupled.
Yeah.
And that's possibly due to the majority of people being vaccinated, and they're just very small outbreaks that are happening.
And it looks to me, when I look at the numbers, that we're at the limit of detection, and there's certainly no evidence within the wastewater of a huge spike at this stage, I think, despite its variation. Thanks, Jennifer. There's moral sort of distress and et cetera have caught the attention of the public. Marguerite is wondering if perhaps there could be decreased empathy for healthcare workers who are suffering sort of a moral distress. And then Joe wonders if people such as yourself, or people who oversee residents themselves who have a moral stress, do they actually develop moral stress themselves? So two rather existential questions. I'm not sure. Yeah.
Okay. Decreased empathy is associated actually with something we call burnout. Yes. Physicians suffer burnout a lot, like more than half of physicians under normal conditions. That's sort of emotional exhaustion, depersonalization, and a decreased sense of sort of accomplishment. In essence, what that means is like feeling less connected, I guess, to the patients that you're treating. And I think that's definitely a concern, especially where there might be really high patient loads, where they just keep coming. And I think we might see that more early in the pandemic, where a lot of, well, I guess it's still true, but in places where a lot of patients died and there was a lot of death and a lot more sort of feeling like there was nothing you could do, because there was at the beginning like a lot treatments were not effective. Nobody knew what was going on. So I do think that is related to moral distress, but probably also related to burnout. I'm mentioning a lot of different concepts that kind of overlap, none of which are sort of a mental health diagnosis, much like we might say stress, which can mean different things. The second question, the people who oversee residents are largely practicing physicians that are sort of supervising. So absolutely, they can be suffering from the same condition. It can actually probably be worse because they're often the ones making decisions. So what needs to happen with these morally distressing sort of environments is, as much as possible, organizational sort of strategies, taking any decision making that's problematic out of the hands of doctors. So if there's a need to triage, to decide who gets resources, to have really strict kind of protocols in place that make it so that you follow the protocol and you're not making personal decisions.
You're not owning it.
That can help. I don't know how much that helps, but
sure.
Yeah.
Are there other interventions, Jennifer? It sounds like such an intractable problem. Are there other things that one can do just to reduce?
Rotating staff between high and low stress roles. So not having all the nurses working in the ICU forever, trying to rotate them out to other places. A problem that we've seen in the pandemic is redeployment. So healthcare workers being moved to do jobs that they're not adequately trained for. That's really challenging. There's individual interventions. Again, these are sort of like self-care. It's a little problematic because it puts the onus on the healthcare worker that's already completely stressed and burned out and doesn't have time to do these things. Again, I just stress that the evidence shows that the organizational changes, as much as possible, are more important and better to intervene against these situations.
Is there, I mean, if there is somebody in the audience, for example, who's feeling right on the cusp right now as far as stress goes, and this is perhaps not fair to you, Jennifer, is there a resource that you can direct them? I'm sure there's tons of them. But
well, actually, at the Royal, so I work at the Royal Ottawa Mental Health Centre, we have the Prompt Clinic, which takes referrals. We also have Frontline Health. I can look it up and try to put it in the chat, but there's a specific service available actually for healthcare workers that they can access resources sort of confidentially through that service offered through the Royal as well.
Good to know. Okay, Jennifer, thanks very much. Ruth, I think we've answered most of the questions, and I think we've covered most of everything. Roger, just tell us what you just put in the chat there, just to end up.
One of my pediatric emergency colleagues and one of my dearest friends' husband truly struggled with all the stuff that Jennifer talked about, and he just wrote a very courageous essay in the CMAJ, which is a Canadian Medical Association journal, talking about his own suicidality. Very courageous. He's an adult emergency physician. She's a pediatric emergency physician. Very courageous, very eloquent, very powerful, and highlights exactly everything that was talked about earlier. So I think it answered some of Joe's questions and even ties into what Marguerite was also referring to, and how it's important that another thing to bring up. There's a great book. That's an essay. If you want to read a great book, Jillian Horton is an adult internist based in Manitoba, and she writes often for the Globe and Mail, so to the lay audience, and has also written a book called We Are Perfectly Fine, which is a bit of tongue-in-cheek, meaning that we are not, and that's her response. How are you doing? I'm perfectly fine.
Yes.
But it's a memoir of her struggles with coping with some of the stresses and hazards of bringing on a lot of the patient suffering. So wonderful. A great memoir, powerful essay of anyone interested in going into the healthcare field.
Okay, and Jennifer's put that into the chat as well, the Frontline Wellness. Thanks very much, Jennifer. Tyson, Jennifer, Roger, wonderful job. You made me look good, and that's the important thing. Great talks, all of you. Ruth, over to you.
Right. Well, thank you, everyone. I'd like to thank all the speakers, Roger, Tyson, Jennifer, for the great presentations, super informative. And I'd like to thank our moderator, Alex, for the highly interactive discussion. You know, sometimes we don't realize it, but it really brought to light the impact that this pandemic had on healthcare workers, who are really the heroes through all of this. So we really have to thank them. This entire session has been recorded and it'll be available on our website. So if you missed part of it, it'll be out there. I'd also like to thank the University of Ottawa Brain and Mind team that worked to herding cats and getting everybody together. Our manager, Natasha Hollywood, administrative coordinator, Candice Fortier, and our science writer, Sarah Schock. And also, if you'd like more information on this or attend our future events, please go to the University of Ottawa Brain and Mind website. Follow us on Facebook, on Twitter, and please join us for tomorrow evening's event using artificial intelligence to explore the frontier of neuroscience. So again, thanks everyone for the great discussion, and thanks for joining us tonight. Bye.